All right, sounds good. Well, then what I want to do today is so you've probably gotten into the rhythm of this class up to this point. It's basically we have these discussions. There are videos to watch. There's an exam to complete, and sometimes assignments to hand in, and it's basically going to be that for the rest of the semester. Um, some weeks are going to be heavier. Some weeks are going to be a little bit lighter. I try to space things out like that so that it's not just the exact same thing every single week. I would say, though, that this past week was probably one of the heavier ones, though, especially for those people who are probably not as into bio or as excited as about bio as I am. Hey, Hope, welcome. So I do want to spend just a little bit of time making sure that we are all on the same page, that we understood what happened in over the past week, because these are concepts we're going to be using for the rest of the semester. And it's going to be important that even though you've already taken your exam and hopefully you are here prepared and ready with that information, with that knowledge, that we reinforce it. Hey, Athena. All right. Okay, now that a few more people are here, one thing, we start promptly at 1 o'clock. Please be here at 1. But otherwise, any questions or concerns about the past week's content? Okay. So here's what I want to do. Before we get into the content from this past week, I want to revisit creating experiments. We practiced this a little bit last week. I want to do this again, just because the more we do this, the more second nature it's going to become. And in fact, later on in this lecture, we're going to start to understand why doing these kinds of things over and over again makes it more second nature. My goal by the end of this class is that if someone says, I want you to write a hypothesis, you're going to know precisely how to do that. And if someone asks you to design an experiment, you're going to know each of the steps involved in doing that. So to help with this, I'm bringing this question. I think this is a pretty cool question. It's a relatable question for a lot of people, I think, because most people have pets that they might want to train to do something. But how about this? Are some breeds of dog smarter than others? How would we go about experimentally testing this question? How would we go about experimentally testing whether some breeds of dogs, whatever breeds you want to talk about, are smarter than others? Because I think we all at least maybe have an intuition that this is true, right? There are some dogs that seem smarter. How do we do that?
You could get a couple of breeds together, like different breeds, like one is a poodle, the other could be a pit bull, the other could be a German Shepherd, and see how and test how long it takes to train the different breeds of dogs.
Okay, perfect. So in that scenario, what is the independent variable?
The type of dogs.
Right, the type of dog or the breed of dog. So we're working with poodles, we're working with pit bulls, and we're working with sorry, what was the third one you said? Labradors, Labrador retrievers.
It's a German Shepherd, but it could be like anything, I guess.
Yeah, and it could be anything. German Shepherd. I like those. That's perfect. So our independent variable. How do we define that again? What is an independent variable? Why is the breed of the dog the independent variable?
Because I don't know. I guess it's depending on. Oh, thank you.
Go ahead, Hope.
Oh, I said it's what you change.
Okay, so it's the thing that we, as the experimenters, the people who are actually conducting the study, are going to change. Now, another way to think about it is the thing across which we want to compare something. So here we want to compare how smartness or intelligence or the speed of training differs across different breeds. We're comparing different breeds. I want to make a very important note here. If we were to define our independent variable that way, that we're just going to bring dogs in of different breeds and then compare them, do you see any problem with that when it comes to as an experimental design? Does anyone see a problem with that? So we design an experiment, we bring in 3 different breeds of dog, and then we divvy those 3 different breeds of dog into different groups, and then we compare them. Is there anything that doesn't really conform to an experimental design about that?
Is it because it's too biased?
How so? What do you mean?
Well, I say like different breeds of dogs are some breeds can be smarter. Like they say the poodle is pretty smart versus the pit bull, which I mean they're smart in their own way, but they could be like I've seen like many a hyper pit bull before. But I guess it's just biased because depending on like breeding, um it you know breeding and what kind of dog they were bred for, that could be also very different. I
True.
guess it all depends on how they you know what they were bred for, really.
Yeah, and I guess that's kind of our question, right? Is we think that these breeds are going to be different. I guess these are all that's all really good points. What I'm trying to one thing that I think about here, and I think that everyone needs to keep in mind, is that when you do a truly experimental design, what you want to do as the experimenter is you want to directly control whether one group receives a treatment that the other group does not. The thing about bringing in different breeds of dogs that are already a certain type of breed is that you're not making them a certain type of breed, right? They're already coming into the experiment that way. When someone or something comes into the experiment already a certain way, what that's called is a quasi-experimental design. Quasi just means like fake. So it's like a fake experimental design. It's still technically an experimental design, but there are certain things that you can't assume because you're right, it's biased. It's biased because we didn't put them into those different breeding groups. Can you think of a way that we could get around that, where we're actually manipulating breed?
Maybe like breeding them yourself so that they're all raised in like the same environment.
Exactly. Yeah. You would actually have to be the one who creates the breed, right? You'd actually have to pick the mother and the father and have them reproduce and create offspring, and then that would presumably be of a particular breed. You are controlling that. So for the sake of this, let's just say that we do that. One way or another, we're breeding the dogs together, we're creating those breeds, we're causing the difference between them. I only emphasize this because you'll be tempted to do this probably for your own experimental designs. I have a lot of people give me quasi-experimental designs. A good example of that would be like measuring personality, for example. A lot of people might say, oh, I want to compare people who tend to have a lot of anxiety versus not a lot of anxiety. They experience a lot of stress or they don't experience a lot of stress. Technically, that's an experimental design because you're comparing 2 different groups, but it's quasi because people are already coming into the experiment either anxious or stressed out or not. How would you compare people who are stressed versus not using a true experimental design? How would you, as the experimenter, manipulate how stressed someone is? Make someone stressed or not? How could you experimentally manipulate how stressed someone is?
You find out like what makes like triggers them to make them stressed and maybe either do that. Like a test or something could make someone really stressed.
Right. Maybe not figure it out ahead of time, because if you figure that out ahead of time, then you might accidentally only be using things that are stressful for some people but not others. But yes, something like a test, maybe like, or something that maybe would be stressful to everyone would be something like, maybe put them in a room where there's this sound, there's this like staticky sound, or like really whiny, like high-pitched sound that's just constantly playing, and they have to sit in that room for a while. After a while, I think that would stress most people out. So you could put them in a group where either they have to sit in that room with that sound or not. That would be a true way to manipulate stress. But if you were to bring people into the experiment who are already either stressed or not, that's not a true experiment. That's a quasi-experiment. So remember that. Gotcha. Sounds good. Thanks, Michael. All right. So let's go back to our breed example. We're manipulating the breed. Let's say we're breeding them one way or another. We're getting poodles, pit bulls, and German shepherds. That's our independent variable. Okay, what's our dependent variable here?
Would it be the tests that you're putting them through?
Right. However, we're measuring smartness. So how would we measure how smart a dog is?
You could, you know those little contraptions that like the dogs can get food out of, and you can measure it like that.
Yeah, so what exactly would you be recording? What would you be measuring about those puzzle boxes? I think they are.
How long it takes for them to complete the puzzle box.
Good. That would be a good operationalization, an operational definition of how smart they are, right? Because it's a number that you can record. It's how long, how much time has passed before they finally open the puzzle box. Good. I like that. Other ways that we could measure smartness or intelligence of an animal. I want to practice doing this a few different ways, because there's never usually one perfect solution. For example, with a puzzle box, one of the things that could happen is a dog could be simply quicker at opening a certain kind of puzzle box because it's already opened something else that's like that puzzle box. So that's not necessarily a test of its intelligence. It could be a test of its experience with stuff like that. So is there any way that we could kind of get more directly at intelligence?
We could have them do like a little obstacle course.
Yeah. What would we record? What's our dependent variable there?
How long it takes them to get through the course.
Perfect. The amount of time it takes to get through the course. Yes. I had a student in another class who was talking about how you can train certain dogs, certain hunting dogs, right, to respond to, say, a gunshot. When they hear a gunshot, that should cue the dog to say, "Oh, there's something may have been shot. I should go chase it down and see if I can retrieve it," right? So when you're training a dog, usually what happens is you have to go through a few trials, right? Where, there's a gunshot, then maybe you tell the dog, "Hey, you need to go fetch that now, or you need to go find that." You need to direct them in some way. So you over time, you train them to respond to the sound of the gunshot. So another way that you could, say, operationalize that is how many trials it takes for them to learn that they need to go fetch something, they need to go retrieve something when they hear a gunshot. Something like that too. That would be a good measure of intelligence. Any other ways you kind of want to throw out there for how we could define intelligence?
So you could do like 3 cups and you could put a treat under one and make sure the dog's watching and see if they can figure out which cup it's under.
Oh, interesting. That's almost like measuring perception, right? It's how closely can they follow. That's a good one. That is a kind of intelligence, right? It's kind of like being able to maintain focus. How well can you maintain focus? I like that. So then what is it? What is the number that you're recording? That would be your dependent variable.
So it would be maybe how long it takes for them to figure out what cup it's under, or maybe how, I don't know, I think I said the right thing.
No, you're good. You could do the amount of time, but maybe it's not really the time it takes that we want to know, right? It's what? How often they do what?
How often they get the treat. Well,
Yeah,
figure out what cup it's under.
right, how often they actually, yeah, how often they actually make the right decision, right? So maybe what you do is you actually put them through several trials. Maybe you do this cup game like 10 times, and then you count up how many times they correctly guessed out of 10 times. I like that a lot. That's a good one. That's clever. Okay, so that's our dependent variable. So now what we can do is, so we've operationalized the independent variable. That's going to be the different breeds. We've operationalized the dependent variable, either the amount of time it takes for them to pick up a behavior, how long it takes them to get through a maze, how many correct times they guess where the treat is underneath the cup. Okay, let's assume that our two variables are the breed and the number of times they correctly guess where the treat is under the cup. What would be our hypothesis? And remember, this should always begin with, I predict that. What are we predicting about the relationship between the independent and dependent variable? Let's say our 3 breeds are poodles, pit bulls, German shepherds.
Well, while all dogs have a highly developed sense of smell than humans do, I predict that the German Shepherd will have a much higher sense than the pit bull or the poodle.
A much higher what?
Since they can smell like better. I don't know. I think they could just pinpoint like smells better than other breeds. I mean, why would cops use like German Shepherds as the drug dogs, you know?
But remember what we're measuring here. We're not measuring sense of smell. We're measuring
Oh,
intelligence. How are we measuring
intelligence. Yeah, I don't know.
How are we measuring that? How did we decide to measure that? It was the cup thing, right? So what are we recording? We predict that German Shepherds will
Probably pinpoint where the exact like location of the item in the cup based on smell.
Yes, and more often they'll do that, right? So if we're giving them 10 trials of, here's putting a treat under a cup, where is it? And then they guess, and then we do it again, and then we do it again, and we do that 10 times. We're going to count up the number of times they get it correct. So that's our dependent variable. So the prediction is going to be, I predict that German Shepherds will correctly guess where the treat is more often than the other breeds. Or another way to say that is, if we're saying that the number of times they correctly guess it is their score on this little cup test that we're giving them, that the German Shepherd will score higher on this score of, they'll score higher on this test. This level of detail is important because the hypothesis should include the operational definition of your variables, right? I think that this particular breed is going to score higher on this test, or I think that the German Shepherd will take less time to complete a puzzle than the other breeds. Always make sure that you put your operational definition in the hypothesis. That's important. Okay, so then from there, when you write say this is a hypothesis that you're really excited about, you then write a research proposal. You'll then run through exactly how you're going to say recruit the dogs, how you're going to breed the dogs, right? What breeds you're going to compare. You're going to describe that cup task. So you'd actually have to describe, okay, 3 cups are going to be put down. A treat is going to be hidden underneath one of them, et cetera, et cetera, et cetera. Run through exactly what each dog is going to be asked to do. Describe that I'm going to do this 10 times. They're going to go through 10 trials. And then I will count up the number of times they correctly guess, where the treat is. Something like that. So that's what you're going to have to do for the proposal. All right. Okay. What I want to do is I want to give everyone an opportunity that if you have a hypothesis that you feel pretty good about or that you're not really sure how you'd make an experiment out of it, I want to take time to do that right now. So if anyone has an idea and they kind of want to work through it right now, let's do that. I'll say this. If it is your idea and we work through it in class, you may use that idea for your research proposal. Or if you haven't yet done it for a hypothesis, you may write it for your hypothesis. That's your incentive to participate.
So I already used this on one of my hypotheses, but my question would be, does social media use correlate to happiness?
I like that a lot. Okay. How would we experimentally test that? Let's run through the steps. What is the independent variable? What's the dependent variable?
Well, the independent variable would probably be the person that you're testing, and the dependent variable would be like how much social media they use, probably.
Well, not the person. So if we want to know how social media use causes a change in happiness, what is the independent variable there? It's not the person. And other people, you can help out. It's a collaborative effort. If we think that social media use is going to cause a change in happiness, what's the thing that we want to manipulate?
Would it be the time spent on the social media?
Yeah, right. It's the social media use itself. So how could we divide people up into different groups based on how much, say, social media they use? How do we make that experimental group, the control group, etc.? If we're going to experimentally manipulate the amount of time spent on social media, we have to divvy people up into groups, and they're going to receive different experimental interventions. How are we going to split people up into those groups? What groups are we going to create?
You'd have to randomly assign, right?
Good, but what would those groups look like? So it's 2 groups, 3 groups, 4 groups. What's the difference between those groups?
I guess you could
Have
do
like
you could do like 3 groups, and one of them being having no social media, the second one having maybe like 30 minutes to an hour of being on social media, and then the other one spending hours on social media.
Perfect. Yes. So in that case, you have two experimental groups, one where it's a little bit of social media time, 30 minutes to an hour, versus hours. When you say hours, always make sure you specify precisely how much, because when you put people in these groups, you're going to be saying, okay, you're going to spend 3 hours on social media per day, something like that. Try not to keep it open-ended. But yes, the basic design is, yeah, yes, that's correct. One detail we'll need to specify is when you say 30 minutes or an hour, is that per day? Is that per week? That's another thing that we make sure to specify. Whenever you're thinking about time, you usually have to have some kind of reference. Hours per day or hours per week, etc. Okay, and then there's the control group, and that's important. I'm glad you caught that, where they have to not be using social media. Could we add a placebo group? And what would that look like? What's a placebo again?
A placebo is like a fake version, kinda.
So could we have a placebo group for social media where they're still doing the kinds of thing that someone on social media might do, but they're not actually on social media? The reason we want a placebo group is because if someone is just not using anything, then they actually differ in two ways from the experimental group. They differ not just in how much time they're on social media, but they also differ in that they're not doing anything. So in this placebo group, what we want to do is we want another group where people are doing something. They're just not on social media. So what could we have them do that would be like being on social media, but without the social media? Or they'd be doing something. It just wouldn't be social media. Maybe we could have them, oh, go ahead, Bree.
They could like be texting somebody or FaceTiming.
Okay, yeah. In that case, you're doing something that is socially interactive. It's just not the same formula as social media. You're not like scrolling through posts. You're not actively clicking on stuff. You're not, yeah, that could work. I'd like that. So maybe you just have someone having a conversation. Or maybe have them, I don't know, write a personal blog, right? So maybe some people spend time on social media by like posting memes or actually like posting something that, you know, their thoughts, right? So they're posting their thoughts. It's just that they're not posting it publicly for everyone to see. Something like that. Still can't hear you, Michael. All right, so that would be our independent variable. Now, our dependent variable, happiness, how are we measuring that?
By maybe like how often they show like signs of happiness, like smiling or laughing.
Yeah, good. I like that. So then what's the number that you're recording?
So the number of how often they give like a positive facial expression.
Good, perfect. The number of times they smile, the number of times they laugh. Or, you know, since it's happiness, we could also talk about sadness, the number of times they gasp or they cry, or, you know, we could get We get creative. The number of times they feel angered or outraged, which is probably more appropriate for social media. That's fine. Yes, you can type. That'd be work well. Okay. So that's one way that we could record happiness is we actually we observe what someone is doing, their behavior, right? We count up the number of times that they smile. One thing that I suggested for a couple of people is that you can actually do something like a 1 to 10 scale for something. So if it's something straightforward like happiness, you can usually do this. I don't recommend getting into a habit of it though. And I'll tell you why. Scales are good when the thing that you're recording is easily accessible to the person. So happiness, how happy you feel in a moment, actually is pretty accessible. You know how happy you feel, usually. But if you start to think about something like, if you want to say, if you're trying to measure memory, some people don't always have a very clear understanding of what they remember and what they don't. For example, you could ask the question like, does the amount of sleep that you get affect a person's memory? And you could ask someone, how clearly do you remember X, Y, and Z on a scale from 1 to 5? The thing is, though, a lot of people don't. They might think that they remember something when they really don't. One thing about sleep, and we'll actually learn about this later in class, is you need a particular type of sleep to it's called to consolidate a memory, to actually strengthen a memory overnight. And so some people think that they can get 4, 5, 6 hours of sleep and be totally fine the next day. In a lot of ways, you can be. You can do certain things on very little sleep. But in terms of memory, your memory will be worse the next day if you don't get a particular type of sleep called REM sleep, which is you only get in a big chunk near the end of a night's sleep, around hour 7, 8, 9. So people can feel like they have good memories even though they don't. In those kinds of instances, it's not very good to use a 1 to 10 scale or a 1 to 5 scale because they don't have good access to that. So think carefully about that. Scales are fine to use, but be careful about them. Okay, so let's say that's our experiment. So now we've divvied people up into different experimental groups where they either get social media, they Skype with someone. They don't get social media, they Skype with someone, they spend 30 minutes on it, or they spend 3 hours on it. And then we're going to record the number of times they smile or get angry, or however we want to measure that. Okay. We've identified the variables, we've operationally defined them. So what's our prediction? Eli, since this was your idea, what's the prediction here?
That more use of social media does affect happiness, and the people who use it more won't be as happy.
Okay, good, but use the operationally defined variables. Use the numbers. How are the numbers related to one another, or how are the groups? So say people who are in this group will people who are in the groups where they spend more time on social media will do what? How are we recording happiness?
We'll score a lower score on the happiness rating or whatever we were doing.
Okay, we could use a happiness rating, or what was the other thing we said? We're going to count up what? What would be like an observable or a behavioral indicator of happiness?
Like number of times seen smiling.
Exactly, yes. So the prediction is people who are in the experimental groups where they are asked to spend more time on social media, they will smile less often than people in the control group. If we think that social media is going to make you less happy, something like that. Would anyone predict the opposite, that the more time you spend on social media, the happier you would be?
honestly just depends on like who you have on social media and like who your friends are, whatever, etc.
Yeah, probably, right. Whether you're like, whether you're following certain people and that's what, kind of fills up your news feed. Yeah, yeah. All right. Is that clear? So you'd have to describe that entire design. You'd have to use those details. Make sure that you operationally define your variables. That's super important. If people get points off, it's always because they don't use those operationally defined variables. All right, anyone else want to throw an idea out there? I am happy to spend time on this. This is so important.
Okay, so makeup and clear skin.
Okay, yeah, okay.
Okay, so the independent variable would be the makeup used, and the dependent variable would be the skin, clear skin.
Okay, good. So how are we
So-
operationally defining each? Good.
So for my experiment, my dog is squeaking her toy. I'm so
I
sorry.
see that. Don't worry about it. Don't worry about it.
So we'd break them into groups. So clear, probably females, but I mean males can wear makeup too. So put them into 3 groups. You could do people not wearing makeup, people with maybe a little bit of makeup, like just foundation or just concealer and mascara, and then you could do the 3rd group, which is like a full face of makeup. So like foundation, all of that. And so we are going to manipulate how much makeup they wear, and to see how their skin is. So we're going to measure how clear or how not clear their skin is going to be with makeup or without makeup.
Good.
So
How
we-
would you
we can-
measure? Yeah.
So we can
Keep
measure
interrupting.
that by maybe seeing if they have any acne, aging, etc. So how much they can. Sorry, I'm gonna take this
No,
toy
you're,
from her.
no, you're doing well given how distracting squeaking can be. No, you're doing well. Good, good, good. So if we're gonna quantify acne, we'd literally count what probably?
By how many like acne they have, how many pimples they have, or blackheads, or etc.
Good, good. When you say aging, that's a good one. How would we quantify the age of skin?
How many wrinkles they have. If they have bags under their eyes, we can count all that.
Very good. Okay, good. Perfect. That works very well. Those variables are correct. They're operationally defined well. So what is your prediction?
So I predict that the amount of makeup you wear will make you have more acne.
One small change about the beginning. So not just the amount that you wear, but you think that people who wear more, right?
Right.
So people who wear more makeup will
Have more acne or
to be really precise, they will have more pimples. They will actually.
right. Okay.
Right. Yes. Yes. Because acne is more of like a generic term. Yeah. Okay. Good. Perfect. Then you just have to describe that in a research proposal. So yes, you can submit that as a hypothesis. That is your research proposal now. Anybody else want to practice? All right. What we'll probably do is do this at least one more time. I think next week I have something like this planned. So if you don't have an idea now and you want to do this kind of practice in class where we all help each other out, bring an idea next week. All right. I'm happy to spend time on this. For now, let's move on. So in this past week, we learned about the biology of psychology. We learned about neurons specifically. And there's a bit of information I want to make sure we all understand very, very clearly. You probably do, because hopefully you studied for the exam and you did well on it. But in case you're kind of forgetting stuff, this is information we're going to use again and again and again throughout the class. So what does a neuron look like? Who wants to generally describe a neuron for me? What are the major parts? What are those parts responsible for? What does the neuron do? Just kind of give me your overall impression of what a neuron is.
A neuron is a cell that transmit information from the brain to like a part of the body or vice versa.
Perfect, yes, it's a cell. And that cell is specialized for sending information from some part of the body to another part of the body, or transmitting information about the outside world into the body, right? Okay, good. So how does it do that? It does look like a worm with crazy hair. So it does have, what are those crazy hairs? Describe these structures for me.
Those are the dendrites.
Okay, good. So the dendrites do what?
They receive information from outside the cell.
Good. So they're going to detect something from outside the cell. Now, it could be that, it's you pressing something against your skin. It could be that there's another neuron communicating with that neuron. Either way, the dendrites are going to bring that information into the cell. Okay. Once it's in that cell body, that kind of central area with the nucleus and all that, you then have that long strand coming out of it. What's that called?
The axon?
Good. You have this axon. That transmits what? How is that information sent? When we say it communicates information, what actually is being communicated?
Some electrical impulse.
Good. An electrical impulse. And yeah, what is that called? What's that other name for that impulse?
Synopsis?
Not
No?
that's at the very end. What's the
Ooh.
electrical impulse called? It's an action potential. I'll use that term. I'm going to interchange neural impulse or electrical impulse. I'll say that sometimes. Sometimes I'll say action potential. Those two things are synonymous, all right? But I want you to know each. It's an action potential. It's that spike in the electrical recording where the cell becomes really, really positive. Okay, so we're sending that electrical impulse, that action potential, down the axon. Then at the very end, what's at the end of a neuron? What's that very end part called?
That's a synopsis, right?
So it's going to, the synapse, not the synopsis, but the synapse, that is the
I was
space.
pronouncing it wrong the whole time.
Yes, so it's a synapse. The synapse is going to be that space in between the end and the next cell, whatever cell that neuron is trying to communicate with. So that's the space. But what's that actual end part called? What's the very end of the axon called?
The axon terminal.
Good. And what's contained in the axon terminal? What is the neuron storing there? What are those little chemicals that are released from the axon terminal called? They're packaged together into those kind of little packets. Neurotransmitters. That's going to be a real important term.
Uh,
Neurotransmitters. Yeah, neurotransmitters. That's stuff like dopamine, serotonin, endorphins, acetylcholine. There are going to be a few of these that we talk about and reference again and again throughout the semester. So it's important to understand when you get to the very end of that axon, you're sending that electrical impulse, and then once it gets to the end, that electrical impulse causes neurotransmitters to be released. So that's why you're sending that electricity in the first place. That's why we even care about that. It's really to tell these neurotransmitters to release. Okay, once they're released, they are released into the synapse or the synaptic cleft. Those are both terms you can hear, synapse or synaptic cleft. Once they're in there, what they're going to start to do is bind to the next cell. So if that neuron is trying to communicate, it's trying to send a message to another neuron, or it's trying to send a message to a muscle cell or a liver cell or your pancreas or your heart. When your heart pumps, the only reason it pumps is because there are neurons telling it to pump. So it releases those neurons bind to the next cell, and there are receptors on that next cell that they bind to. And from there, the next cell does something. Okay, I want to emphasize that what happens at that next cell is going to depend on what neurotransmitter is released. So I already mentioned this. Some neurons are excitatory, that is, they cause something to be more likely to do something. So if acetylcholine as a neurotransmitter, when that's released, it makes a muscle more likely to contract, more likely to kind of say, if you talk about the muscles in your arm, to pull that arm back towards you, contract your muscles. There are also types of neurotransmitters that make your muscles less likely to contract, to relax. Those are inhibitory neurotransmitters. So the electrical charge is just simply saying whether those chemicals get released, but what those chemicals are will determine what the action is, what actually happens because that message was sent. So we can talk about a few things here. If we talk about serotonin, that is a mood regulation neurotransmitter. It makes you calm. I'll give you this example. When you're really stressed out, let's say that you have a big test coming up, and you feel really stressed. Your heart's pounding. Your mind's going a mile a minute. You're kind of upset, and maybe you're getting a little bit sick, and your stomach's kind of churning a little bit. That's stress. You take the test, and then when it's over, you experience maybe a little bit of relief. But when you get that test back, and then you did really well, it just kind of leaves you, right? That feeling that you get when that stress just melts off, that's actually due to serotonin. In that moment, your neurons are releasing serotonin, telling your body, okay, we're no longer in this stressful situation. We no longer have to worry about this. So, turn off that stress now. People who have trouble regulating their anxiety, they feel stress often and maybe too much for their own good, can often kind of maybe not produce as much serotonin as someone who does do well handling stress. So these chemicals and how they're released actually directly affect the kind of thoughts we have, the behaviors we engage in. And I think this is why we need to learn how neurons work. This is why I stress this information. If we understand how that electrical impulse is sent and how those chemicals are released, we can start to understand people's behaviors a little bit better. For example, if we know how much someone feels stress is due to a chemical, then we can design drugs to alter that chemical. And that ends up being what most medications are. If you take a medication that's like an anti-anxiety medication, it's usually a medication that's going to somehow increase the amount of serotonin that's in your synapse. It's going to calm you down more, etc. When we talk about drugs of abuse, becoming physically addicted to certain drugs, usually what's happening is you're altering dopamine. So with alcohol, with cocaine and methamphetamine, with anything that anything that causes a release of dopamine tends to be physically addictive. You can be physically dependent on it. Again, if we know that, if we understand kind of that is because of a chemical, we can maybe, you know, change someone's experience at that level. This is why it's important to understand how neurons work. Okay. So we learned what a nervous system is, and we learned that these are the two things that it does. The nervous system communicates information through this network of neurons and neurotransmitters being released from those neurons. And that's its basic function. Throughout the class, I'm going to be giving more examples of how your nervous system also is responsible for making predictions. So it's not enough that you just communicate information. What you want to be able to do is, over time, maybe try to predict what's going to happen in the future, what might change. When you learn something, this is actually what's happening. When you learn something, your nervous system makes a prediction. Let's say that you're training a dog to be a hunting dog, going back to that example. And so you want it to respond to the sound of a gunshot, such that, you know, when you shoot something, it goes and it retrieves what you've shot. Well, if the dog has never done this before, it doesn't know that that gunshot means something. But through repeated training, what it can learn is that that gunshot predicts that an animal might be down out there that needs to be retrieved. So the dog, over time, starts to use its nervous system to predict that a gunshot means go fetch that. We do the same thing. How does our nervous system make predictions like that? Can you think of any examples like that? How does your nervous system make predictions about what might happen in the future?
You can get like a bad feeling about something, like going
Oh.
to a certain event or place.
What you were probably taught when you were young is that you should probably stay out of dark alleyways, right? Or you should if there's a group of people, be careful about your belongings, right? And so what you've learned is, when you are in a large group, you should predict that someone might try to steal something from you, because it's easier to steal stuff from someone in a group. So your nervous system says, okay, I predict that stuff might get stolen. So what do? You maybe keep your purse closer to you, or you keep your hands in your pockets so that no one can reach in there after it, or keep your wallet in your front pockets rather than in your back pockets, which are easier to pickpocket. You're trying to make predictions about what might happen. That's the role of your nervous system. And we're going to get back to this. We'll talk about a few ways of that when we learn through things like classical conditioning and operant conditioning, which you may have heard of elsewhere. What that is these are all ways in which you learn how to predict that something's going to happen in the future. Okay. Then last time I started to introduce, I believe, this idea of heuristics. And I introduced these as simple, efficient ways of solving a problem, a rule of thumb for doing something. I don't know if you've had time to ponder this a little bit, but we're going to learn that heuristics are really what we're trying to build when we kind of exercise our nervous system. When we are building those connections, where electrical impulses can say release certain chemicals and cause those chemicals to create a certain response, we're building heuristics. A lot of heuristics, I'll just mention this, can either develop early or they're heavily influenced by biology. We don't need to learn them. So there are actually a lot of connections within your nervous system and within your brain that are there when you're born. Or shortly after you're born, they're formed and you don't have to, their experience isn't required for them to form. There just needs to be time for you to actually make those connections. So sometimes they're heavily influenced by biology, other times they are learned as well. When you learn how to do CPR, that is your heuristic. Your heuristic is, if someone starts choking, immediately start doing the Heimlich maneuver. That is a heuristic you've learned. The Heimlich maneuver is not it's not natural. It's not biologically determined. It's something that it's a heuristic you had to build up over time. It's a connection you had to build up over time. But let me give you some examples of things that you didn't have to learn. Those of you who have interacted with an infant for one reason or another, what do you do if you gently touch the bottom of an infant's foot? So if this is their foot and this is their palm is like the bottom of it, you just graze your finger across the bottom of an infant's foot. What happens?
They quickly pull it away because it's ticklish.
Okay, they might pull it away because it's ticklish. What's the other thing they do? So when they pull it away, what happens to the shape of that foot?
It curls. They like curl their toes or either spread their toes out. I've seen both.
Yeah. No, but both do happen, absolutely. But this reflex of it curling up like this, that's pretty consistent. If you do this to an infant's foot, it'll do this, and it'll do it very early on. It doesn't have to learn that. It's not necessarily thinking, oh, there's something on my foot, I better curl my toes right now. This also happens with infants' hands, right? Very early on, if you put something in an infant's hand, it ends up curling its fingers to grab onto it. And then they never let go. Infants have incredibly strong grip. They're surprisingly strong. But why curl the foot? Well, the idea here, I'll just kind of give this to you here. The idea is that this is actually a reflex that's left over from our evolutionary ancestors. So we actually share a common ancestor, an evolutionary ancestor, with things like chimps, orangutans, other primates. And if you look at chimpanzee feet, they look like ours in a lot of ways, but they have really, really long toes. So much so that they can actually grab things with their toes and they can hang on. Now, the idea is at some point in our evolutionary ancestry, we actually lost the really long toes. So we have that reflex that if something grazes our foot, we should grab it. However, because we don't have these really long toes, it doesn't really do much for us. So the idea here is we have this reflex. It does this simply because it's kind of a preserved nervous reaction left over from kind of earlier in our ancestry. Another example I kind of want to give of kind of a heavily influenced biological response, that's something you don't have to learn. It's just something you kind of come into the world with. Let me give you this example. All right, you are you're finding a date. Okay, you find a date online, or maybe you met him at like I don't know. You met him at a bar. You met him at church. You met him at school. Whatever. You find someone, you really like them. They really like you too. You decide to go out on a date together. Maybe you go out to dinner, you get some lunch or something, you have some friendly banter, and it's going really well. You really like them. Everything you suspected that you would like them was confirmed. So you decide, you know what? Let's go back to my place, and maybe we can cuddle on the couch, and we can watch Netflix or Hulu or HBO Max or whatever people watch now, YouTube. And then just kind of, you go back, you cuddle a little bit, you're watching stuff, maybe you kiss, you use your imagination, however far it goes. Afterwards, you're kind of sitting there and you're, in the kind of happiness, and you're just talking about and sharing stories. And, one of you decides to share the story that, I have this kind of story within my family of my grandma, who's just this crazy person, just a crazy grandma. And she did this thing like 20 years ago. It's infamous within our family. Everybody knows about it. And the other person goes, oh, that's so cool. That's so interesting, because actually in my family, we have the same story about our grandma who like 20 years ago, she did this thing, and everyone thinks she's crazy. And then you're sitting there and you're like, hold on a second. What's the name of your grandma? And turns out you both have the same name and you both have the same grandmother. What's your immediate response? If you were in that situation, what would be your response? What would you do?
Panic.
Okay, panic. What else might you feel in that situation? You've been kissing and cuddling this person. Now you find out that you are from the same family. Okay, so there's this kind of panic. Would anyone feel kind of a lot of people end up feeling a disgust response where they might actually go, oh my God, I can't even, and you want to distance yourself from that person. You might even say, you know, I got to get out. I got to get up. I got to go. I got to get out of here. It's kind of a strange response because this actually was someone who you were jiving with, you got along well with, and in every respect you enjoyed, except for the fact that you found out that they were genetically related to you. Why? Why do we have that response? Does anyone know? Why do, and this is something you see in humans and you see in non-humans too, why do we have a disgust response towards incest?
Because our culture has made it unacceptable, but like in old times, like monarchies, that was a common thing.
Okay, so not even just in old times, there are societies now where incest is relatively more acceptable. In the United States, it's absolutely illegal. In certain countries within Europe, it actually is legal. You can get married to a close sibling. But if it were just culture, and there is a very strong cultural component here, but if it were just culture, we wouldn't also see it in other animals. You pretty much see it in any sexually reproducing animal. Do you know why? Is there a problem with siblings having a child together? Or does there tend to be a problem?
There tends to be like more of a rate of like autism and stuff.
Okay, certain genetically heritable disorders or diseases, yes. It turns out if you have sex with and reproduce with someone who is within your genetic lineage, that they are part of the same family, one of the risks that you can run is if within that family you harbor certain genetic abnormalities or genetic disorders, then having those two people mate and produce a child is going to substantially increase the probability that that child inherits those genetic diseases. It can sometimes get like a double dose, which is what you often see in the case of, I don't know if anyone knows within the Jewish community, there's something called Tay-Sachs disease, where if either person has just one copy of the Tay-Sachs gene, not a big problem, or not a huge problem, but if you inherit both copies, say from each parent, then the child has extremely difficult respiratory problems. Most children die from that. So what you can see with incest is you can get something called inbreeding depression, such that the chance of a child inheriting a genetic disease is much greater. So the idea here is that and you see this across cultures, even in cultures where incest is legal, there is usually still a predominant disgust or avoidance response to incest. And you see this in things like, you can see this in mice. If you put a bunch of mice in a cage together, and some of them are siblings and some of them are not, mice will prefer to mate and reproduce with non-siblings before they mate with siblings, generally. There are some exceptions to this, but this seems to be at least the large trend, is that organisms tend to avoid, sexually reproducing organisms avoid incest to avoid inbreeding depression. So that's a heuristic. That's a simple, efficient solution to a problem. The problem is inheriting genetic disease. The simple, efficient solution to that is feel disgust about incest. Yeah, Athena, you're gonna say something.
We're talking about humans, but what about animals? Because I had to get my cat Cammy fixed before her son impregnated her.
So one of the things about especially pets, like pets owned by humans or living within a small community, is that they may not have many mating options. And so in situations where there's really nothing else that they can reproduce with, you'll often see that they'll just look past the whole "you're my sibling" thing. This is why you can end up having, say, if you have like, you know, a brother and a sister housed in the same house, they never see any other animals, and they don't get fixed, they will usually eventually try to reproduce with one another. That's partly because of lack of options. Yeah.
I've also seen it around with all the stray cats around the area that um it'd be, I think we named him Daddy Cat. He was the big strong male cat that ran around the area, and then he was always with this one cat. Always they had, many litters together. But I think that Daddy Cat was possibly my cat Cammy's father. Possibly.
Right. Yep. I mean, cat colonies, you can have some crossover there, but I think especially within kind of small rural areas where you tend to, especially if, people are feeding cats and then they create these kind of small enclosed communities where everyone's basically clustered around the same house, you can very often get siblings kind of producing children together. And non-human animals suffer the same problems with inbreeding depression. You're just running the risk. It's not that if a child is had by incest that it will always be a problem. It's that there's a much greater risk of inheriting a genetic disease. And that's the problem. Okay, so these are other examples of heuristics. Now, remember, when we're talking about heuristics are those connections that we're creating. The connection is if you notice that someone is genetically related to you, the response is feel disgust, feel that I want to avoid this at all costs. So we're talking about a connection here. That's what a heuristic is. So when we're talking about neurons, your body is constantly building and modifying these connections within your nervous system. And the way that it does this, and I realize I'm introducing a new, extremely important term within the last 7 minutes of class, but I want you in your notes to star this term big time. I will be talking about long-term potentiation again and again and again and again. Long-term potentiation refers to the fact that when you stimulate a neuron, by stimulating it, you strengthen the connection that neuron forms with the next cell. So if a neuron is connected to another neuron, if you stimulate that first neuron, it's going to form a stronger connection with the next neuron. All right, that's long-term potentiation. Very important definition I want you to know. What does this look like? So here's what we know about neurons so far. This image right here, what you see here is the axon terminal. This is where those neurotransmitters are. Here's the synapse in between. Here are the receptors that accept those neurotransmitters. And then here's that next cell, whatever it happens to be, another neuron, a muscle cell, etc. Down here, I apologize that there are no X and Y axes, but here's what this is showing. This is a graph of the strength of the electrical charge that these neurons will create. So let's say that we, in this initial state, stimulate the neuron. It creates a certain electrical charge that then tapers off. That's what we're measuring here. Before repeated stimulation, there might be a connection. But when you stimulate that neuron over and over and over and over again, that connection strengthens because of two reasons. And if we go to the right here, you'll see why, or see how. One, the more you stimulate a connection and you cause neurotransmitters to be released, the more neurotransmitters this neuron is actually going to create. So you can see the big difference between here and here is that there are more neurotransmitters. The other difference is these are the receptors beforehand. After long-term potentiation, after repeatedly stimulating it, you now have more receptors. So you have more neurotransmitters and you have more receptors. What this causes biologically is you're going to release a ton of neurotransmitter now after long-term potentiation, and that ton of neurotransmitter is going to be received by a lot more receptors. What that creates is a much more, it's a much more substantial electrical charge. So this thing, whereas before you were kind of getting this strong of an electrical charge, now this electrical charge is much stronger. Okay, so long-term potentiation is when you stimulate this connection over and over again, you're going to get more neurotransmitters, more receptors, and that causes a stronger electrical response. This is learning. This is how learning occurs. When you create a connection in your brain and say you are trying to remember the parts of a neuron, what you're doing is you're trying to put the neurons that are in charge of your memory of what an axon is, you're trying to stimulate that connection over and over again so that when you hear the word axon, you immediately know, ah, that's that long structure in a neuron. That's a connection within your brain. Another way to think about long-term potentiation is it's the principle of use it or lose it. That is, the more often you use a connection, the stronger and faster it becomes. And if you don't use it, then it becomes weaker. If you play a sport, let's say you play basketball, the first time you learned how to do a layup, you probably had to really think it through. It was not quick. It was not a confident response. Maybe you kind of had to, okay, and then I put my arms this way, and then I push the ball up that way, but you don't know where you need to actually like send the ball. And so over time, you practice how to do a layup. The more you practice it, the more automatic it became. The easier it was to do it correctly. That's long-term potentiation. You're stimulating those neurons, those connections responsible for your ability to do a layup correctly and quickly. That's also why if you don't practice things like that for a while, you can kind of lose that ability. So after this class, after you've taken the chapter exam for this chapter, you may not practice and think about what are the different parts of a neuron. And if you don't do that, eventually the connections in your brain that are responsible for your memory of what a neuron looks like, they're going to get weaker. You're going to forget what a neurotransmitter is. You're going to forget what a synapse is, et cetera, et cetera. That's weakening it. You use it or lose it. This is also why I have you practice hypotheses and experimental design. Again, by the end of this class, what I want you to be able to do is if someone asks you to write a hypothesis, you know how to do it. And if you need to design an experiment, you don't have to sit there and think about it and go, okay, now I have to identify the independent. What is it? Which variable do I identify? Just automatically, okay, identify the independent variable, identify the dependent variable, operationalize those variables, end up creating a prediction, randomly assigning participants to different groups. You should be able to run through that process like nothing. You're trying to stimulate those neurons over and over again. All right. Does long-term potentiation make sense? We're going to go over it again and again and again. This is not the last time. But make sure you study that. Keep that in mind. Keep stimulating your connection, your storage, the connection you have for long-term potentiation. Here's what I'm going to leave you with. Here's a critical thinking question. I've taught you about cognitive biases. I've taught you about heuristics, and now I've taught you about long-term potentiation. These are all related somehow. Take some time, think about this. We will revisit this during our next discussion and try to figure out what is the connection between them. If you can figure it out, you're keeping up. All right, this week there are videos posted for the nervous system. So we're getting away from the microscopic stuff and we're now looking at the bigger picture, how the nervous system in general works. We're going to talk about the endocrine system, which is your hormone system, which actually is very similar to your nervous system. And then we'll talk about the brain, which of course is a very specialized part of the nervous system. All right. Thank you all for being here. I am out of time. Everything should be uploaded. If you have questions, please do let me know. And I will see you next week. Thanks, everybody.